Hello everyone, welcome back to class 12th chemistry. Question for you: what's the one molecule that carries the instruction manual for your entire body? It's nucleic acids. Today's we'll study nucleic acids from NCERT chapter 10 Biomolecules. We'll understand what they are, the difference between DNA and RNA, and structure of nucleotides, all in exam focused language. If you are preparing for CBSE, ICSE, NEET, or other state boards, this is a must know topic. Let's start. Every generation of each and every species resembles its ancestors in many ways. How are these characteristics transmitted from one generation to the next? It has been observed that nucleus of a living cell is responsible for this transmission of inherent characters, also called heredity. The particles in nucleus of the cell responsible for heredity are called chromosomes, which are made up of proteins and another type of biomolecules called nucleic acids. Nucleic acids are mainly of two types: the deoxyribonucleic acid, in short DNA, and ribonucleic acid, in short RNA. Since nucleic acids are long chain polymers of nucleotides, so they are also called polynucleotides. Chemical composition of nucleic acids: complete hydrolysis of DNA or RNA yields a pentose sugar, phosphoric acid, and nitrogen containing heterocyclic compounds called bases. In DNA molecules, the sugar moiety is beta D2-deoxyribose, whereas in RNA molecule it is beta D-ribose. DNA contains four bases: adenine, in short capital A; guanine, in short capital G; cytosine, in short capital C; and thymine, in short capital T. RNA contains four bases. The first three bases, adenine, guanine, and cytosine, are same as in DNA, but the fourth one is uracil, in short U. Structure of nucleic acids: a unit formed by the attachment of a base to one dash position of sugar is known as nucleoside. In nucleosides, the sugar carbons are numbered as 1 dash, 2 dash, 3 dash, etc. in order to distinguish these from the bases. When nucleosides is linked to phosphoric acid at 5 dash position of sugar moiety, we get a nucleotide. Nucleotides are joined together by phosphodiester linkage between 5' and 3' carbon atoms of the pentose sugar. The formation of a typical dinucleotide is shown in figure. A simplified version of nucleic acid chain is shown below. Information regarding the sequence of nucleotides in the chain of a nucleic acid is called its primary structure. Nucleic acids have a secondary structure also. James Watson and Francis Crick gave a double strand helix structure for DNA. Two nucleic acid chains are wound about each other and held together by hydrogen bonds between pairs of bases. The two strands are complementary to each other because the hydrogen bonds are formed between specific pairs of bases. Adenine forms hydrogen bonds with imine, whereas cytosine forms hydrogen bonds with guanine. In secondary structure of RNA, single strand helix is present, which sometimes folds back on itself. RNA molecules are of three types and they perform different functions. Types of RNA are named as messenger RNA, in short mRNA, ribosomal RNA, in short rRNA, and transfer RNA, in short tRNA. Biological functions of nucleic acids. DNA is the chemical basis of heredity and may be regarded as a reserve of genetic information. DNA is exclusively responsible for maintaining the identity of different species of organisms over millions of years. A DNA molecule is capable of self duplication during cell division, and identical DNA strands are transferred to daughter cells. Another important function of nucleic acids is the protein synthesis. In the cell, actually, the proteins are synthesized by various RNA molecules in the cell, but the message for the synthesis of a particular protein is present in DNA. That's it. So, to wrap up, nucleic acids store and transfer genetic info, two types DNA and RNA. They are made of nucleotides, sugar, phosphate, nitrogen base. DNA is double strand, has thymine, stores genetic code. RNA is single strand, has uracil, helps in protein synthesis. These are the exact point you need for CBSE, ICSE, other state boards, and NEET MCQs. Quick trick to remember: DNA double thymine deoxyribose and RNA single uracil ribose. For board exams, draw the neat structure of a nucleotide and label the three parts. That's a guaranteed 2 marks. If this helped, hit like and save the video for revision. Drop a comment if you want me to solve NCERT in text exercise questions on nucleic acids. That's nucleic acids done. See you in the next one.